This lecture is on pentose phosphate pathway, also known as hexose monophosphate pathway, as well as phosphogluconate pathway. All three names are indicative of the intermediates found in the pathway, and it has been named in that manner. In terms of pentose phosphate pathway, if I am to give any exam question, I will refer it as simply as pentose phosphate pathway. On a whole, pentose phosphate pathway is relevant in generating NADPH as well as the production of ribose. In terms of production of ribose, it is the residue relevant in making the sugar part or portion of nucleotide as well as nucleic acid, RNA or DNA synthesis. Then you have in terms of NADPH, it is a reductant. This NADPH is useful in many of the biosynthesis pathway, such as in the making of fatty acid, in the formation of cholesterol, one of the steroids, as well as in making amino acids. In particular, one reaction that involving the enzyme glutamate dehydrogenase. It's also useful in terms of safeguarding the cell from highly reactive oxygen species with the regeneration of glutathione in red blood cell as well as other cells. And this pentose phosphate pathway, in terms of where it is active in most cells or tissues, except in non-lactating mammary gland, as well as very low activity in skeletal muscle. So here you can see all the three names given for this pentose phosphate pathway, also known as phosphogluconate pathway, also known as hexose monophosphate pathway. It refers to the either the ribose sugar or the 6-carbon sugars present in the pentose phosphate pathway. This pathway can be separated into two, that is either the first phase, which is oxidative phase, or the second phase being non-oxidative phase. The oxidative phase mainly will produce NADPH in two reactions found there. And then you have the second phase, which will produce the ribose 5-phosphate. But apart from the 5-carbon sugar, it also generates other sugar molecules, which has 4-carbon, 10-carbon, 7-carbon, 6-carbon, as well as 3-carbon sugars. And then in terms of its location, it is in cytosol, and in terms of its whether can be categorized as catabolic or anabolic, it is mainly categorized as anabolic. However, it can also play a catabolic role, as you will see later on. So just like any other biochemical pathway, this pentose phosphate pathway also broken down to phases. It's broken down to two phases, that is oxidative as well as non-oxidative. In terms of the whole pathway, it is relevant in generating NADPH as well as ribose 5-phosphate. The oxidative phase has the rate limiting step for the whole pathway itself. That is the first step, glucose 6-phosphate dehydrogenase enzyme. And then the oxidative phase has 3 steps that has 2 steps of it forming NADPH. The non-oxidative phase will generate the ribose 5-phosphate as the most important intermediate compared to the other intermediates, such as the 3-carbon glyceryl diphosphate, the 6-carbon fructose 6-phosphate, the 4-carbon erythrose 4-phosphate, the 7-carbon pseudoheptulose phosphate, and then you also have the 10-carbon xylulose 5-phosphate. And then you also have these enzymes, trans-ketolase, trans-aldolase, where they able to connect between glucose to glyceraldehyde 3-phosphate as well as fructose 6-phosphate. If you recall, remember, these intermediates such as glyceraldehyde 3-phosphate and fructose 6-phosphate are in fact intermediates of glycolytic pathway. So meaning this pentose phosphate pathway is in fact connected to glycolytic pathway by these two intermediates. So here I give you the summary of the pentose phosphate pathway, where its function not only mainly for the production of NADPH as well as converting hexose to pentoses, but also although not the main function, is also to perform complete oxidative degradation of pentoses, where the 5 carbon sugar can also enter glycolytic pathway via pentose phosphate pathway once it forms the fructose 6 phosphate as well as the glyceraldehyde 3 phosphate. And also the NADPH, which is the main function of pentose phosphate pathway, it has many functions. This reductant is able to be used in several biosynthetic reactions such as fatty acid synthesis, amino acid synthesis, as well as cholesterol synthesis. You also have the NADPH serving as a biochemical reductant for the removal of highly reactive oxygen species. You also have this NADPH quite required in the liver cell, where this P450 monooxygenase system is found in order to react with xenobiotics such as alcohol consumption, excessive alcohol, as well as certain degradation of drugs. And then it's also useful in converting the ribose sugar to deoxy ribose sugar of nucleotide, where the C2 prime carbon is removed of the OH to be added with hydrogen instead. So these are all the function of NADPH. In terms of conversion of hexose into pentoses, of course, apart from NADPH, it also makes ribose 5-phosphate, and this is used in forming the component of ATP as well as other coenzymes, the sugar part of these coenzymes itself, as well as nucleic acid.